everybody, welcome to a longer edition of our coverage of the MASAI Trial. And I'm more than happy to have as a special guest here, first author, principal researcher, Professor Kristina Lång from Lund, Sweden. And yeah, thanks for stopping by, Kristina. And let's jump just right in.
Being a clinical researcher in breast imaging is you have to make an effort not to address the issue of using AI. Because there's been some amazing tools available on the market for a few years. And we've done retrospective studies that have seen the potential to use this. But in order to see how it really works in the clinic, you have to do a prospective trial. And the best way to evaluate new clinical methods is to do a randomized trial. And so the trial was designed with the primary outcome of interval cancer rate, because I wanted to see whether AI could improve the efficacy of screening, and then interval cancer rate is the best surrogate measure that we have. And since there are rather few interval cancers, you have to have a large study population. So we determined that we needed 100,000 women in order to address the issue of interval cancers. We also wanted to see whether AI can reduce the workload in mammography screening. And that was, so there are two research questions that we wanted to address. So either workload reduction and if it can improve the efficacy.
Tremendously important research questions, because workload, resources to cover the workload are obviously limited, particularly in screening. This is of utmost clinical importance. And let's just jump into the methods, because this is a broad question. So how did you address this? How did you design the study to meet these questions?
Yes, so the randomized design, we did a parallel design. So there were an equal number of participants in both arms. Mm-hmm. It was a 1-to-1 randomization. So that means that we basically randomized every woman that attended screening to either AI-supported screening or to the control arm, which were double reading without the access of AI. And by doing the randomized design, you can filter away factors that might influence the result. So you can have two populations that are as equal as possible.
Listening to you, Kristina, sounds like an easy to be done exercise, but obviously this is extremely difficult. This is why you have been one of the first research teams who did that. So just share some behind the scenes experience, Kristina. How did you manage to do that? Because setting up a randomized control trial, yes, it's the best way to do, but it's super difficult. And this is why there are only two. There are one actually on this topic. So some behind the scenes stories.
Yeah, behind the scenes. I think, I was just on a conference in the UK. And I got the question, how long does it take to get an ethical approval in Sweden for the MASAI trial? And I estimated that it took about 2 months to get an ethical approval. So I think, and they were really amazed by that because it takes so much longer time in the UK. And I think to have efficient system that we have in Sweden that you can apply to an ethical review authority, you do an online application, the system works really well. So that was not a problem to write and get the ethical approval. But of course, there's a lot of paperwork before starting a trial. You have to do to register the trial with the clinical protocol and to have all these paperwork in place. But I think one thing that made it maybe easier was that we had a really small team working on this. So it's basically myself and my statistician that made all the design and the paperwork. Of course, I had colleagues that I could discuss issues with, but we were a small and efficient team. So I think that was one factor that made the work quite easy. And also, I was rather amazed that many of the collaborations that I had, for example, with Sectra, the PACS provider, and with ScreenPoint that provides the AI algorithm Transpara, and also Unilabs, which is the the breast imaging unit where I work. All these different stakeholders were really positive and easy to collaborate with, and we solved many issues during the process in a really efficient way, I think. One problem that we had was that this was during the pandemic, and when we had some issues with our server where we had the AI algorithm, we had some issues that we need to solve. We couldn't fly in the people from ScreenPoint, and so there were some problems that we had during the process of the trial. But I would say overall that it was a small and efficient team and great collaborations.
Beautiful. This is really amazing inspiration because, yeah, it's not easily done. With that, we will step to the results. Kristina, we know that you started in April, I think, 21 with recruiting. Just how long did it take to the preparation phase? How long did you work on that? It was during the pandemic, so it was obviously in the year 2020. But how long did it take before you recruited or included the first patient?
You mean from start of the design and
exactly everything? From the initial idea?
It took 1 year from idea to 1 year and 3 months from idea to trial start. Yeah, I would say that.
Wow, amazing! This is impressive. And with that, let's just jump right into the results section because this is obviously the most interesting part because the results are spectacular and. Very interesting to discuss. So, first, let's summarize it quickly, Kristina. Choose your focus wherever you wish, and let's chat a little bit on that.
Yes, absolutely. So, with the first results that we assessed was to study early screening performance measures and the effect on workload, because we wanted to make sure that the cancer detection rate did not decline when doing AI-supported screening, since the majority of the women in the intervention arm would undergo single reading. So we focused on the cancer detection rate. We found that the cancer detection rate did not decline. On the contrary, we detected 20% more cancers with AI-supported screening without affecting the false positives. And we could reduce the number of screen reading. So the screen reading workload was reduced by 44%.
And I think this is so important, so we can repeat it. So basically, we even detected a little bit more cancers with AI. I think this is important because it's about safety, but it was even more than that. The PPV was even a little bit higher, if I recall correctly.
Yeah, so there were a little bit more cancers of those that we recall from the intervention arm. Yes.
Beautiful. And the workload, well, that's the idea. The motivation of using AI was reduced. Could you just summarize, like in a practical way, not like in the way it's written down on the paper, but practical? How does this work, or how this will work in clinical routine according to your vision and experience?
AI-supported screen reading protocol that we used was based on AI as a triage tool, so it can identify exams with a high risk of malignancy. And there was about 7% of the screening population, and those underwent double reading. And the remaining examinations underwent single reading, so those were deemed to be low risk. And then we also had the aid of computer-aided detection mark that highlighted suspicious findings in the images. And that was from the 30% most intermediate and high risk. So from risk score 8 to 10 using the Transpara risk score, which divides the screening population in about 10 equal parts.
Okay, that's a beautiful summary. And for those of you who are interested, and I'm pretty sure most of all will do that, we will also link in the bio a beautiful animation summary movie where this workflow is illustrated. Let's go, Kristina, to something what I'm always very interested, what you also very interested, obviously, interval cancer rate and the biology of cancers detected, because it's not only about detection, absolute number, but it's also about the kind of cancer to detect. Yes. So what's your take on this?
What's my take on this? Well, so we detected 20% more cancers with AI, and that involved both invasive and in situ cancers. We could see that there were relatively more T1 invasive cancers, so small invasive cancers, and in situ cancers in the AI-supported screen reading group. It seems like AI can aid us to detect subtle cancers. Now, that could be a good thing, that we can improve our early detection in screening, but it could also imply that the use of AI can lead to an increase in overdiagnosis. I believe that it's both, that it aids both in early detection of cancers and can lead to overdiagnosis. I think on an anecdotal level, I think that we all, I experienced it several times during the trial, and my colleagues also, that you were surprised when you put on the CAD marks and it was shown in the image. Oh my God, I missed that one. So I think it really can, the use of AI can help us reduce overlooked findings. But the opposite was also true, that AI marked many findings that we could clearly say that that's benign. And one of the advantages that we have is that we can compare to prior examinations and see that this small calcification cluster has been there for a long time. It's not a cancer, or it's a calcification cluster that we have already assessed and determined to be benign. So there are different possibilities there where a human radiologist or a human in the loop can assess. But it's an advancement of the AI algorithms also, that they can start to compare with prior. So that's a development of the AI tools. So we will see if this can increase the performance of the AI algorithms further and to reduce the false positive marks in the mammograms. I think so. I think they will be better. We will see.
And then that's a very important point for those of us who are listening and maybe not that familiar with the literature and the status quo, that this is not considering the priors, the results, right? Which is, I think, super important if we practice breast radiology, breast imaging and screening in particular. And it absolutely makes sense that this will be better if you consider the priors. So now we have talked about what drove you to start this huge project. We learned a little bit about the methodology behind. And once again, this is a sneak preview, nothing more, but we learned a lot of that. And I think the key results are clear now to everybody. So from your side, being a clinical breast imager, a researcher, and the principal investigator of this paper, what do you expect to happen at your practice the next years?
It's the near future and what's going to happen in 5 and 10 years. But in the near future, I first of all, we can say based on our randomized trial that it's safe to use AI in screening, since it will not compromise cancer detection. But the study is still limited since it's a single center study and it's performed in a Swedish screening program and on a Swedish population. But another prospective study that was performed in Sweden, the Screen Trust CAD trial, it also saw some similar results, that it was had an increase. That when results from other trials come out, we will see that these results all point in the same direction, that we can actually increase the accuracy in screening. And so that's one thing. So I think based also on the lack of breast radiologists, which is really acute in some places, then since we can say that the AI-supported screening can be considered safe, we could start using AI instead of sustaining double readings, since it might not be possible to do that in some sites. But as a researcher, I would like to have evidence that this actually leads to improved patient outcome. So to get the results from the primary endpoint of the MASAI trial to see the effect on interval cancer, I think would be very important in order to make a general recommendation on whether you should use AI or not. But as I said, in 5-10 years, the algorithms might be even better than we are, and then they should be reading all the screening mammograms instead of us.
Thank you. This was a beautiful summary and outlook as well. So I think with that, it's time to close our quick chat. Kristina, Dr. Kristina Lång, Professor Kristina Lång from Lund, Sweden. leading the MASAI trial. It's been a pleasure having you here as our guest. And thanks very much for taking your time.
Thanks.
And you guys at home, make sure we're gonna link all the material we've been talking about in the bio, so you just can click and watch all the beautiful animation videos and obviously read the paper. Have a good one. Goodbye.